genomes and chromosomes. So this is kind of starting with the basics, reviewing structure of DNA, talking about how the bacterial chromosome is different from the eukaryotic chromosomes that you've probably heard before, and then how do we copy our DNA, and then a little bit about plasmids, which is fun. So let's jump in. There we go. Got your learning objectives, bunch of key terms, a lot of key terms this time, but hopefully a lot of them are ones you've heard before, and it's just like, oh yeah, I got to refresh my memory a little bit on that. So genome is a term you guys should know by now. It's all of the DNA in an organism. Coding DNA, non-coding DNA, all of that. It defines what an organism is. So it tells us not only what to make to build the structure of our bodies, but how that stuff behaves, when to make those proteins, how to develop the cell. It's all of it all together. It's the blueprint to the house that you end up living in. And with the exception of RNA viruses, pretty much all other genomes are constructed of double-stranded DNA. And hopefully we'll get to talk a little bit more about those viruses because I think it's important for you to take with you going on, but we're going to have to fit that in wherever we can. Now, DNA is the reason, or is the one that we store our genetic info in for a couple reasons. It is stable. DNA does not degrade very easily. It's very strong. It's been able, we found it in permafrost, we found it in fossils. It can withstand for a long time, which if that makes us who we are, we want it to be stable. We don't want it to break down at the slightest touch. But it's mutable. Mutable means it can be mutated. We can make tiny changes to it. And that's important because we need to be able to evolve. The environment is going to constantly change around us. If we can't adapt to that environment, we die. So being able to change it when we need to is great. And it needs to be replicable. We need to be able to copy it so that all of our cells, if we're multicellular, have the same DNA, or I can make progeny. So those are the three really important characteristics of DNA. And the very first genome that was ever sequenced for a cell, because the first thing ever sequenced was a virus, was a bacterial genome. And it was for Haemophilus influenzae. So now we have 35,000 genomes for bacteria that we've sequenced. And then like a couple other thousand for plants, couple thousand for mammals, things like that. Now, I'm going to go through this history pretty quickly because I hope to God you've heard this ad nauseum at this point. And if you haven't, I tried to put a video of me really going through the history. It is at a high school level, but you get the high points. So when we discovered the structure of DNA, it's a great study in science because the experiments and the process that we made all these discoveries is looking at how we do science. So the history of it shows us the process of science as well. The three that we really need to focus on today are Griffith, Avery, McLeod, and McCarty, and Hershey and Chase. So Griffith in 1928 did this really cool experiment. So he took a bacteria that had a rough strain and a smooth strain. The rough strain, the R strain, was non-virulent, meaning it can be given to an organism and the organism lives. Virulent means it's very pathogenic. Then the smooth strain, which is the virulent strain, when given to an organism, the organism dies. In this case, it was mice. What he then did was took this smooth strain, the killing one, and killed it with heat. So he basically boiled this bacteria, took the dead cells, gave it to the mouse, and as expected, the mouse should live because the bacteria is dead. Here's where the interesting thing happened. He took the rough strain, the one where the mouse did not die, and mixed it with cells that were killed from the dangerous strain, and then took that mixture and injected it into a mouse, and the mouse died. So he's saying something is transforming the safe, the non-virulent one into the virulent form. And we just have to now figure out what is the transforming material, the genetic material, basically. So he came up with this idea of transformation, which we will talk about a couple more times throughout the semester. It's where bacteria can just take up DNA from the environment or from dead cells, which is not something we can do. Basically, they go around and they're like, ooh, somebody left that DNA on the ground. I'm going to take that, and now that's part of me. Would be really cool if we could do that, but we can't. Now, he died before he could expand on his research, because the next step was going to be, what is the transforming material? So that's where Avery, McLeod, and McCarty came in. They took his experiment and they refined it to try to pinpoint, okay, what is the material that's causing this transformation? So their experiment was really cool. They took the virulent strain and they mixed it with a bunch of different enzymes. First, they mixed it with enzymes that destroy polysaccharides. Okay, is a polysaccharide, is a sugar moving over. So they put in these enzymes that destroy the polysaccharides. If it is a polysaccharide, we've destroyed it, so the mouse should still live. If it's not, then something else is the transforming material. So polysaccharides destroyed, mouse still died, so it's not sugars. Then they added an enzyme that destroys lipids, mouse still died, so it's not lipids. Did it with RNA, did it with protein, mouse always died. Finally, when they added DNAs, enzymes that break down DNA, the mouse lived. So obviously, because we took away the DNA, now the cell isn't dangerous anymore. The mouse can live. DNA was the transforming material. So now the race was on to say, okay, is that the case for all living organisms, that it's the genetic heritable material? But there was also another point to this too. This wasn't completely conclusive because it didn't deal with inheritance. So we needed Hershey and Chase, and just because this is a point of contention for me, we talk about now that, yes, it's good for women to be in science too. There's a lot of us in this room, more than the men, especially in biology. Women have been in biology for forever. Hershey and Chase, that's Martha Chase. She's a woman. So we've been in biology for forever, and we make really cool discoveries in biology. Okazaki is a woman. The people who discovered the enzymes that, well, really Okazaki was a husband-wife team, but she was the one who got the credit for it. The people who discovered all the enzymes for replication, another husband and wife team. She didn't get the credit this time because he did a little bit more work than her, but still, women have always been in biology, so it's kind of cool to hear these names. All right, so Hershey and Chase. I love this experiment because it's so simple and so beautiful. They found that DNA was the thing that moved when phages infected bacteria. So quick little review. When we talk about the 4 biomolecules, sorry, I want to get a marker that works. Carbs are composed primarily of carbon, hydrogen, and oxygen. Those are the base molecules or elements that make up carbs. Same with lipids. Their base structure is made of carbon, hydrogen, and oxygen. Then our other 2 biomolecules, proteins and nucleic acids, they all start with that same base because these are all organic molecules, and organic kind of means carbon, hydrogen, oxygen based. But proteins add a nitrogen, so do nucleic acids. Proteins also add a sulfur, whereas nucleic acids don't have sulfur in their structure. Instead, they add a bunch of phosphorus for those phosphate groups that we'll talk about in a minute. So we can easily now distinguish between DNA and protein just by looking at which one has sulfur, which one has phosphorus. So that's exactly what they did. They used radiation and they tagged the proteins with radioactive nitrogen, and they tagged the DNA with radioactive sulfur, radioactive phosphorus, I'm sorry. So they let the phages then infect the cells, and they looked to see where the radiation ended up. When it was in the protein capsid, when it was the sulfur radiation, they said that all the radiation stayed with the ghosts, the phages that were empty. But when they tagged the phosphorus, in the DNA, they found that the radiation moved into the cell. Therefore, DNA is the genetic material. So those are the three important parts of the story of DNA that tie back to microbiology. If you want more detail of those, the book does a good job explaining it. There's tons of videos out there explaining their experiments in a lot more detail too. So now, when we talk about the bacterial genome and genomes in general, they vary in size dramatically because we need a lot more genetic material to make something more complex. We need more building material to make a mansion than we do a little shack. So that's the idea. In general, the more simple the organism, the smaller its genome. The smallest bacterial chromosome is 106 about base pairs. That's very, very small. It's actually missing a lot of the machinery that it needs to live, but that particular cell lives almost exclusively inside other cells. So it can steal the machinery from other cells if it needs to. But then these 16,000 ones, they're probably ones that live in constantly changing extreme environments. They might be much bigger. Maybe they form like complex biofilms or complex multicellular structures. So it varies drastically, but you can compare the biggest bacterial and archaeal chromosome size to eukaryotes. 2,900 is the lowest, up to 100 million, sorry. And the human genome is 3 million base pairs. So just basically, the more base pairs you have, the more complex you are, the more stuff is going on. Now, most bacterial genomes are circular. Our genomes and the genomes of pretty much all eukaryotes are linear. So we have other problems to deal with. If you took cell and molecular, you're going to remember a lot of the stuff that we do with our linear chromosomes, like with the telomeres and all that. Bacterial genomes don't have some of those problems. And I want to say this now before we get too much in the weeds. What you learned in cell and molecular about eukaryotes is not wrong. But I need you to kind of turn off some of those parts of your brain for this class, because we're learning how prokaryotes do it. We're learning the OG way of doing all this. Eukaryotes, basically you took the classes in reverse. You should be taking microbiology first, because this is how life began and how life evolved. And then from there, take cell and molecular and figure out where we went from here. So you might need to turn off a few portions of your brain and say, no, we're focusing on eukaryotes, not prokaryotes. Because I noticed a couple people in the class on the exam, when you guys were talking about programmed cell death, you did it perfectly for eukaryotes, not for prokaryotes. So it wasn't apoptosis, it was something else. So just keep that in mind when you're going forward. Okay. Most of the time, it's only one chromosome as well. There are some species, like the Vibrios, like Vibrio cholerae, that's the bacteria that causes cholera. They have two. There's some that have linear chromosomes, like Borrelia burgdorferi, the thing that causes Lyme. And then there's some that actually have a mix. They'll have circular and linear chromosomes. Really crazy. The Agrobacterium tumefaciens is an example of that. Any questions? Stop me anytime today, okay? So, really quick crash course in the structure of DNA. Hopefully this is all review for you. DNA is, when we look at our four biomolecules, DNA falls into the group of nucleic acids. It's just basically like, what flavor of molecule are you? What base structure do you build off of? The monomer, or the building block, or the subunit of all nucleic acids, is the nucleotide. And this is a nucleotide right here. It has three parts. The first one here is the nitrogen base. So this is the nitrogen base. And those have four flavors, A, T, C, and G. for DNA at least. And they're split into purines and pyrimidines. The way to remember this, there's a couple different ways. The first way that I learned was purines are pure as gold. That way you always know the purines are A and G, pure as gold. I was trying to remember the way one of my professors here taught me, because he's from Spain, and the way he remembered it was pura agua, or agua pura. So either way, whatever works for you. And then the pyrimidines are just the other two, cytosine and thymine. So when we look at the structures, the purines have a double ring structure. The pyrimidines have a single ring structure, and that's going to be important. So when we learn the base pairing rules, A pairs with T and C pairs with G. We're always pairing one large with one small base. A double base always pairs up with a single ring base. The middle thing for DNA is a sugar called deoxyribose. So with RNA, at this point, you'll have another hydroxyl group, but we've taken that oxygen away, so it's deoxy for DNA. And then we have a phosphate group, very negatively charged. So DNA overall is a highly, highly negative molecule. Cells like to keep the inside very negative. That way, protons always want to move into the cell and create energy for us. And one of the main reasons why the inside is so negative is because all that negative DNA that we have in there. Any questions about the structure of a nucleotide? Review, right? Please tell me yes. Otherwise, what did your 106, 107 teachers teach you? All right, so how do we connect these nucleotides together to make that twisted helix, the twisted ladder of DNA? They are first joined together with phosphodiester bonds. Do you really need to know what a phosphodiester bond is, chemically speaking? No, I'm not going to make you know that. But just know there's an ester linkage, and in the middle, a phosphate group. So that's why it's diester, phosphodiester. So this connects what we call the backbone of our DNA. The phosphates and the sugars make up our backbone. And it's always between the 5' phosphate group of one nucleotide and the 3' hydroxyl group of another. What do we mean by that? We like to number all of the carbons in organic molecules so we can more accurately say the reaction is happening here. So we start here with 1, 2, 3, 4, 5. So this down here is the 3' carbon, and I'm going to go back to here. That way it's bigger. And it's attached to the hydroxyl group. So this is the 3' hydroxyl end. The other one is right here. This is the 5' end, the 5' phosphate end. I'm going to be repeating this over and over. So if you need to study that, that might be one thing. If you're like, I'm a little rusty on that, put your time and effort into knowing that, because it's going to come up a lot in biology. So the 5' end is basically the end that's leading to the phosphate group of the same nucleotide. The 3' end is where we connect to another nucleotide. Any questions about that? Now, how do the bases interact? Because DNA is actually 2 strands. So it's this strand that's connected at the backbone and this strand connected at the backbone, and they connect together via hydrogen bonds. So hydrogen bonds are not true bonds. This is a very poor way to explain them, but I like to think of them as kind of like magnetic attractions. We're not actually combining the 2 magnets together to make a new structure. They just associate and then break apart again, but they stay their own item every single time, whether they're associated or apart. So with hydrogen bonds, we're having a hydrogen associate with some other slightly negative molecule. Hydrogens tend to be slightly positive because they like to give up their electron. Things like nitrogen, oxygen, they like to be more negative because they like to take the electron. So that slightly positive, slightly negative can form an attractive force, a hydrogen bond, because it's always happening with hydrogen. So that causes our bases to bond in a way that we call complementary. So it's not C to C, it's not A to A. That would be identically. Complementary is the bases that match each other that aren't identical. So in this case, A pairs with T and C pairs with G. And the way they do that is A creates 2 hydrogen bonds with T, because the oxygen on the thymine bonds with the hydrogen on the adenine. And then this hydrogen on the thymine binds with that nitrogen on the purine, on the adenine. With C and G, we make 3 of those hydrogen bonds. So which one do you think is more stable and harder to break? C and G, right. And in the end, temperature can break these associations. One of the reasons why it takes so long to heat up water and to bring water to a boil is because water makes these hydrogen bonds with itself. So we have to overcome the burden of breaking all those hydrogen bonds between the water before the water changes temperature. So if you were an organism that lived really, hot springs down by the deep sea thermal vents that are like boiling temperatures, what do you think most of your DNA would be comprised of? AT bonds or CG bonds? Yeah, so this is one way that bacteria and archaea can protect their DNA. They'll saturate it with more C and G in their genome. Just naturally, they've evolved this way. And it keeps the DNA from what we call melting, or the strands separating. Well, I'll repeat that a couple times as we go. No, just know A and T is 2 and C and G are 3. Yeah. I'm going through this just because the structure might come up later, and I'm a very visual thinker. So when you say it's making 2 bonds versus 3, I'm like, yeah, that's great. But as soon as I see it, I'm like, oh, now I'm going to remember it forever. So sometimes I'll go through pictures a lot more in detail, because if you can explain the picture, you really get it, and it helps people learn better. I can't just read words and be like, oh, I get it now. I need to pair it with a picture. All right. So those bonds come together, and the 2 strands now meet in the middle. But the 2 strands don't just meet in the middle and they're both going in the same orientation. No, that would be too easy. They go anti-parallel, meaning they're parallel to each other, but going in opposite directions. Basically, think of the highway. There's 2 stretches of road going parallel to each other. One of them goes north, the other goes south. Now how do we know that they do this? Because of that 5' 3' carbon. The 5' carbon is on the top over here. If we come and look at the structure here, the 3' carbon is at the top and the 5' is down here. So we know just from that structure that they're moving in opposite directions. And if we tried to switch it and you tried to put the bases together, they wouldn't go together quite as well. Their structures wouldn't line up properly. So physics, to be completely honest with you. So all of bio is chemistry. All of chemistry is just physics. So if you know how elements interact based on the laws of physics, you can predict how the molecules are going to form. And so if we know how much percentage of C, H, O, all that, and we can then take them and crystallize them and do X-ray diffraction and get an overall structure, we can figure out the basics with the chemistry of it all. So unfortunately, I know we don't like to think it, but biologists really do need to become chemists, because the more you understand the chemistry, the easier the biology becomes. Okay, sorry. So at very high temperatures, those hydrogen bonds break. That's why we call it melting. So sometimes if you've ever heard your PI or someone in the lab saying, well, what temperature does DNA melt at? That's what they mean. how does it dissociate from itself? The two strands separate. We also call this denaturing because it technically is. The structure of the molecule is denaturing, just like with proteins. When they change their shape, they denature. That's all that means, is we're changing its shape from the functional to a non-functional state. Any questions about this? All right, still going with the structure of DNA. All right, because of the way that the molecules come together, when the DNA two strands interact, they naturally form this twisting structure. And when you look at that structure, there's a big gap here between the backbones. Here's a backbone, here's a backbone. There's a big gap there. But between these two backbones, the gap is a lot smaller. So naturally, the way it twists, we form a major groove and a minor groove in our DNA. The major groove is wider, the minor groove is smaller. Now, this is important because in the minor groove, it is very, very, very hard to get at those bases. Things won't be able to interact and like bind to specific sequences that are in the minor groove. But the major groove, where the DNA is more open, a protein can come in and chemically recognize, oh, this is an AT, this is a CG, this is another CG, and it can bind to it if necessary. So a lot of our regulators that tell the DNA when to copy, when to not copy, repressors and activators, they recognize these sequences that are open in the major groove and bind to them. Yes. No, that was just me giving an example. It could be anything, could just be all A's and T's. So it depends on the gene, it depends on the organism, it depends on the molecule that needs to come in and bind to the DNA. They all have their own sequence that they recognize. And that allows for specificity. Because if they all bound CG heavy areas, well, then how would I be able to tell it I want gene A, not gene T right now? So you would have to have more specificity than that. So we want it to be more random, unless it's a repressor that we want to repress a bunch of similar genes at one time. And then they would have very similar sequences that the protein would recognize. A's and T's or C's and G's, it just depends. Yep. Yeah. So the major groove, because it's more open, we can have easier access, if you're a protein, to those bases. So if you can see the bases and bind to them, you can regulate the DNA. The minor groove, it's very tight. The proteins have a hard time getting in between the outer area, the backbones of the DNA, to get at those bases. So we don't tend to bind there, because we can't get the specificity that we want. It does happen, but we're more looking at binding the major groups. Good question. Okay, ready? Cool. So another interesting thing, just like with our cell membrane, how we had the top and the bottom are pretty hydrophilic and charged, and the middle was pretty nonpolar, hydrophobic, kind of the same with our DNA. The bases are fairly hydrophobic structures. So the inside of our DNA molecule, where the bases come together, is fairly hydrophobic. The outside, with those phosphate groups, very, very negatively charged, very hydrophilic. So there's another hurdle that proteins have to kind of pass to get to the DNA and bind to it. The proteins will have to be similar to membrane proteins, where they have portions that can adhere to the hydrophilic parts of the DNA and the inner hydrophobic parts. So this just adds to the complexity. Not only does the protein have to be hydrophobic to associate with the bases in the major groove, it also has to have the right structure to bind the correct code within the major groove. So everything is super specific with DNA, which is nice because we don't want to, put a bathroom where the kitchen should be as we're building our house. All right. What else do I want to say about this? That's it. Yeah. Any questions? So it's kind of old, some new with this, right? You're hearing it at a deeper level, I hope, already a little bit. If not, good, you had good teachers. Okay, so here's a picture of this. Here we have the DTXR protein from Corynebacterium diphtheriae. It's the cause of diphtheria. And what this is we call it a dimer because it's two proteins that function together to block access to the DNA. They're repressors. If they're there, nothing else can get to the DNA and copy it or make protein. So they're repressing the function of that DNA. We'll talk more about that later. But they're interacting in the major grooves with the different code of DNA in those major grooves. There's also parts that we can't see here where it interacts in other ways with the minor grooves, but the majority of the points where it attaches are in the major grooves. That was the point there. And you'll hear this a lot. A lot of our repressors and activators function as dimers, where two of the proteins come together and clamp the DNA on either side. Di just means two, mer means unit, so it just means a 2-unit repressor or a 2-unit activator. There's one that my lab studies where the protein itself is a dimer where two proteins come together, and then it's a dimer on top of that, so we call it a dimer of dimers. There's crazy bio stuff here. I'm not gonna quiz you on that. Okay. Any questions here? All right. So RNA and DNA are different. They are both nucleic acids, just like lactose and glucose are both sugars. The same idea. They fall under the broad category of sugar, but they're different molecules within there. So the ways that RNA differs, it's usually single-stranded. It contains a ribose sugar instead of a deoxyribose sugar in its nucleotide. And the base uracil replaces thymine. So it behaves just like a T, but instead of T, we have to write U. Do we remember this? Okay, good. Now, we tend to depict RNA as single-stranded, but the majority of the time it'll fold back on itself and form these really complex, cool structures like you see in the bottom right here with hairpin loops and lots of fun branchings. And that gives the RNA the ability to function also as an enzyme. So it can catalyze reactions because of the shapes that it can take. You don't have to know the details of that, but we'll hit that again later. Good? All right, moving on now. That was the structure of DNA. Let's talk about the important stuff now. Not that that wasn't important, but. All right, so the bacterial chromosome is highly compacted. So just like ours, we compact them into those little cute X shapes, and then we move them around during mitosis. We always have to organize our genetic material, or it runs the risk of getting broken or damaged, things like that. So this is actually a real picture that's been colorized of DNA coming out of a bacterial cell. And you can kind of see, it's a little hard to tell, but it seems like it's coming out in these loop structures. And that's exactly how bacteria like to organize their DNA. So overall, we're going to talk about E. coli a lot because it's the model organism for bacteria. We know it in and out. It's highly studied all over the world. And from here, we can get the basic information and then check other species with it. So E. coli is about 4.6 million base pairs. It's a pretty big bacteria. And this is a really small space to pack that into. Overall, if we were to stretch out the DNA just like with us, it's a lot longer than the cell. It'll stretch out 1,500 times longer than the cell itself. For us, take the DNA out of one of our cells, and it's 6 feet tall. So it's taller than me. About 3 to 4% of the weight of bacteria tends to be DNA. But how do we pack it in? They coil it, as I just said, into what we call domains. These are just a series of loops that they connect together with histone-like proteins. We use histones to wrap our DNA around. These are just histone-like proteins. They're not histones, they're similar. And when we pack it all together like this, we call that area where it's organized the nucleoid. Now, on top of forming these loops, the DNA itself is under twisted or over twisted. So we like condense it even more, or we pull it a little bit further apart, and we call that supercoiling. Whether it's negative or positive, we just call it supercoiling. Because super just means above. We've coiled it, and now we're coiling it in a different way again. And so these green are the histone-like proteins. One of these loops is the domain, and within there, we have various levels of supercoiling of our DNA. And we're going to talk about supercoiling a bit. Ready? Okay. Because we supercoil the DNA, it creates a unique topology. If you've ever looked at maps or anything like that, the topology of an area is its surface structure. There's mountains over here, there's a valley over here. So we can tell where we are and where we're going because of the topology. So there's two types of supercoils to create this topology in DNA. We have positive and negative supercoils. Positive is overwinding it. We're adding more coils, positively adding more coils. Negative is underwound. We're going to remove a coil. We're going to make it less tightly compact. So most cells, their DNA is in the negatively supercoiled state. Why do you think we would want to keep our DNA negatively supercoiled in an underwound condition? Saw a hand gesture there. Why do we want it less wound than tighter wound? Yeah. Easier to kind of take apart for replication. So it doesn't get tangled. Okay, all great ideas. Yeah. So basically, we negatively supercoil it so we can have access to it to do stuff to it. If it's super coiled, it's like that minor groove on steroids. We're tightly packing that down. Nothing can access the DNA and figure out what the sequence is to be able to bind. But when we underwind it, it's a little looser. We see more of those bases peeking out, and we can get in there and grab them and attach to it. And it's easier to open up the DNA to copy it or open it up to make mRNA and proteins. So yeah, you guys were all right to some extent. So they're easier to separate. Now, archaea, because they got to be different, are often extremophiles, often living in really high heat conditions, really high acid conditions that will denature the DNA as well. What they do, rather than keeping their DNA negatively supercoiled, they will keep it positively supercoiled because they're worried about their DNA falling apart. So they'd rather keep it more tightly wound and unwind what they need as they go because it's just safer to protect the DNA. Alrighty. So how do we introduce these supercoils? So usually it's 3 steps. 1, we have to break the DNA. There's 2 different ways that we can do this. You can introduce a double-stranded break or a single-stranded break, and we'll talk about the single-stranded in a moment. So here we take our chromosome and we cut both strands at the same time. We make a blunt end. Then we take one end and pull it through that cut that we made. And then we reattach it with the broken ends going kind of on opposite side, like over the loop that we just pulled through. So it kind of makes a figure 8 now. And you might be thinking, well, it's just a loop. You take that figure 8 like a hair tie. So I cut it, I pull it through, and then when I'm done, it's now in a figure 8. And you're like, well, I could just go like this. Yes, you can, but now you've added an extra twisting into the DNA, so it's not going to be twisted the same way it was when you started, because you've introduced another coil. So when you open it back up again, there's an extra turn in there, or less of a turn, depending on which way you go. This is very hard to visualize. I get that. There's a lot of cool videos out there. The textbook itself has some good videos. I've recorded one so far. I'm going to record the other two and post them for you guys today when I get home. It just took a lot longer to do that than I was anticipating the other night. All right. So when we supercoil, we're changing the topology of the DNA. So things can interact with the DNA differently when it's supercoiled negatively or positively. So we use topoisomerases, the enzymes that alter this supercoiling. The topoisomerases alter the topology. So it's really topoisomerases, but everyone says topoisomerases. Now, there's two different types of topoisomerases, and they'll introduce different types of supercoiling. And so we have to balance their actions. We want one to do one job, one to do the other. So we kind of have to direct when they work. And we'll talk about that more too. So type 1 topoisomerases, this is usually one single protein, not a complex of many proteins working together. These ones make single-stranded breaks. When we cut just one strand of DNA, we call it a nick. What that does is it creates a gap in the DNA, and it kind of creates torsional or twisting stress that the cell now has to relieve that twisting stress. And so what this does is relieves supercoiling. So if we want to negatively supercoil, or we positively and we want to bring it back to a neutral state, we use these type 1 topoisomerases. You can see this DNA strand here has 5 coiling points right here. When we nick it and we do all this work, the resulting molecule only has 4 coiling points. It's more relaxed. I have a hard time visualizing this too. I won't make you explain to me how this works. You just need to know that type 1, they cleave one strand and they relieve supercoiling. They make it less coiled. One second here. I think there was one more thing I wanted to say. Nope. Okay. Then the next kind are type 2 topoisomerases. These are going to be complexes. So multiple proteins working together. What they do is they're the ones that cut both strands. They make that double-stranded nick, and then we do the pass-through like I just talked about. So the one that does this, you've probably heard it, is DNA gyrase. Instead of calling it type 2 topoisomerase, we say it's DNA gyrase. So we use this guy to introduce more coiling, to positively coil. And these guys are a target for antibiotics because their DNA gyrase in bacteria is slightly different than ours. So slightly different. So sometimes these antibiotics also affect us, but not nearly as bad as for the bacteria. One of those are the quinolone antibiotics. You've probably heard of those before. You've probably even taken one like ciprofloxacin, Cipro. These are given often for malaria because they treat helminths as well and things like the little malaria worm. They are not fun to be on long term. I was on one for a month when I went to Africa, and it was made my stomach not so happy. All right, because it's killing all the bacteria in my stomach too. It's just killing all the bacteria. Any questions about that? All right, so this is the video. I think you guys might be able to access it directly from the slides. If you can't, I recorded a screen grab of it and I posted it on our class page. And I'm going to do that for the other 2 videos in here too. I just haven't done it yet. So let's get into replication now. How do we copy our DNA? We have to do it accurately, and we have to do it quickly, and we have to coordinate it with septation, the dividing of the cell like we talked about before. So in the vast majority of species, DNA replication is semi-conservative. And I say that because literally make any rule in biology, there will be something that's like, no, I'm just not going to do that. There's just rebels everywhere. So semi-conservative model says that we take the parental DNA, we separate the strands, and those each serve as a template. And so the new DNA comes in and copies itself using the old as a template, and then they just stay stuck together. So the new DNA is 50% old, 50% new. You've heard this before, right? That's the Meselson-Stahl experiment that also used radioactive particles to figure this out. They're fascinating. If you want to learn more about that, look them up. They're these cool old guys now. All right. Let's jump in then. This is fun stuff. So with bacteria, they always begin their replication at what we call the origin. The gene name for it is ORI-C. So we're going to take a quick little pause here, and I'm going to explain one thing, because it's going to come up. You're going to see these different ways that we write things, and I want you to understand it. When we talk about genes, they're always lowercase to start, and they're always italicized. So the gene for the origin is ORI. It's always 3 letters that are lowercase followed by a capital letter. Proteins are a little bit different. We start with a capital letter. So in this case, let's do another example just because we don't turn the origin into a protein, so there's no matching protein. I worked on a protein called Tol-C. That's the gene. The protein for it would be written like this: non-italics, capital letter T, everything else is the same. So if you see different things, you're going to see a DNA K, that's a protein. If I wrote it like this, DNA K, that's a gene. If you know that and you take bacterial genetics, Dr. Takahashi is going to think you're smart. All righty. So the gene name of the origin is ORI-C. This is the part that's stuck to the membrane. Remember we talked about how the chromosome is anchored to the membrane of bacteria? This is the anchor point. So after initiation, we form a replication bubble. We just split the strands, and it looks like a literal bubble in the DNA. From there, we create 2 different directions that we are replicating the DNA that we call replication forks. One of them is going this way around, the other goes that way around. So we cut it open, and we move in opposite directions towards the other side of the cell. And we're going to end 180 degrees away from the origin, all the way literally on the opposite side of the genome, because it's all a circle. So we call this replication bidirectional because it's going in opposite directions away from the origin. You've heard this before, right? Sort of, maybe. All right. When we get to this opposite side and we stop replicating, it's called termination. And we look for a terminator site in our DNA that tells the replication machinery we're done now. And then we end up with 2 copies of our genome, which is what we wanted. Then they can separate and the cell can septate. So we're going to dive into this a little deeper. And we're going to talk about exactly what goes on in initiation, elongation, and termination. Now, before we move on, remember that once the origin is replicated, these 2 new origins move to opposite sides of the cell, just a little bit, not a lot. They're not in the center anymore, and they link to the membrane a little bit apart from each other. Once that happens, the septum can begin forming. After a brief refractory period or waiting period, those 2 new origins can start to copy themselves even before the cell has finished dividing. So this can happen pretty quickly, and that allows bacteria to double very fast. All right, let's dive in. Here are all of our enzymes that we need to know. So these are all in the protein form with their names. DNA A is the only one that you have to know the protein name of because we don't have a fancy name for it. The rest of them, I'm 100% okay for you to use the common name for them and not their protein name. So DNA A is the initiator protein that's going to recognize the sequence at the origin and bind to it. DNA B is helicase. You guys have heard of that one, right? It unzips the DNA, unzips your genes. That's the joke. DNA G is primase. It will make the RNA primers. Okay, I do need to hear this from you guys. You can like keep it hidden if you'd prefer. How many of you have heard that DNA replication needs primers before? Yeah? Okay, most of you? Cool. I'm just figuring out how much time I need to spend on everything right now. All right. DNA polymerase 3, or DNA Pol 3, we often just call it Pol. This is the major replication enzyme. It's the one that's going to do the copying, add in the new nucleotides. DNA polymerase 1 is going to say, okay, now that we're done, this primer, that's RNA, needs to come out, and I need to put DNA instead, because we don't want RNA mixed in with our DNA. And finally, we already mentioned this guy, DNA gyrase, our topoisomerase 2. It's going to be ahead of this whole replication fork. So the replication machinery is right here. It's going to be further ahead, relieving the torsional strain that it's creating. Because the unzipping, if you've ever tried to take a string that's attached to something and unwind the strands that make up the string, you know, as you pull them apart, it just becomes a twisted mess the closer to the anchor point. So DNA gyrase kind of relieves that strain. Are we ready to dive into this now? It's fun. So a note on DNA polymerase, because it's got to be weird. DNA polymerases, their full name is DNA-dependent DNA polymerases, meaning it needs a DNA code to work from. Some viruses have RNA-dependent polymerases, DNA or RNA polymerases, meaning they can copy it straight from RNA. But in pretty much all cells, all of their polymerases are DNA-dependent. They can't do this from an RNA template. And they also have one more issue. Just like with us, we can't read a book or a sentence from right to left and understand it if we were to read each letter individually. Obviously, we can read the words and we're smart, we can piece it together. But if we were to read a book backwards, it would make no sense to us. Same for them. They can only work from the 5' to the 3' direction. They can only write the new code in that direction. And it's because of their chemistry. So they need a free 3' OH group because that's where they catalyze that phosphodiester bond that connects the backbone. So if they don't see a 3' end here, they're like, well, I have nothing to make that first connection point, so I can't even start this whole process. So they need that to be able to move forward. If they saw a 5' end, they're like, well, I can't do anything with that. All right, so that means DNA is always made 5' to 3'. And that's going to cause some problems later, and that's why we need the Okazaki fragments. Okay, any questions about those? So let's start with initiation. This is the start of DNA replication. When we start replication, that whole timing is controlled by DNA methylation, which is really cool. So we add methyl groups to the DNA, and I think I've mentioned that DNA or bacteria do this before. It's how they recognize this is my DNA versus virus DNA, because viruses don't add methyl groups to their DNA. So as we have DNA existing in the cell, it'll start to add those methyl groups. And that, how do I want to say this? Sorry, I'm trying to think of the easiest way to explain this to you guys. Once we know that the DNA is methylated, then we can add all of our proteins and bind them to the origin. It won't bind, all these proteins won't bind unless the DNA is methylated. If the methyl groups aren't there, it won't even attach to it. So we attach at the origin, which is a 245 base pair sequence that has a series of repeated sequences, and that just helps the proteins recognize this particular sequence. If we start replication at the wrong time, so an ill-timed initiation, we're going to have problems with dividing the cell. We don't want to start it too soon or too late, because then if it divides, it can cut a growing chromosome in half, and now nobody has a chromosome. If it's too early, then we've got too much DNA taking up energy and taking up space in the cell. So we prevent inappropriate initiations with this protein called Seq-A. And what Seq-A does is it binds to hemimethylated DNA. So when we first copy the origin, that new DNA, half of it hasn't been methylated yet because it's brand new. We've had no time to come in and put our methyl groups on it. So Seq-A notices that one strand is methylated and one strand is not, and it binds there. And that prevents all the replication machinery from coming back in and restarting replication too soon. So it kind of gives us a refractory period, a waiting period before we can start to copy again. What will then end up happening is the proteins will come in to add the methyl groups, and they'll kick the Seq-A off. And then that signals to the cell, okay, we've done what we need to do, you can start replicating again. So we have Seq-A bound to just newly replicated DNA because it's only hemimethylated. And then as the cell catches up to, oh, crap, we've got new DNA that's made, I got to go methylate it, then we can kick it off, and then the cell knows, okay, it's safe to start another round of replication. And all initiations are triggered by DNA-A, the initiator protein. This guy comes in, binds to the origin, and starts to recruit all the other proteins that we're going to need to do this whole mess. And DNA-A, when we first divide the cell, its concentration is really low. So it takes a while for it to be copied and that protein to build up enough in the cell to be able to bind to the DNA and start replication again. So we have a couple different ways of keeping replication from happening too fast after we just did a round. DNA-A concentration and that Seq-A. Any questions, or do you need me to re-explain any of that? All right. So when it's bound to the origin, it's in a form called DNA ATP. So the DNA-A has ATP attached to it. And it's going to help recruit what we call the replisome, all of the proteins needed to do replication. And the DNA starts to melt, the strands separate, and that creates the replication bubble. And in that bubble, DNA-C, which is the helicase loader, takes DNA-B, which is helicase, and it loads it onto the single-stranded DNA. So we separate it. Some guy comes in and says, here, I'm going to help you, and sticks helicase onto the single strands. Then DNA-B, helicase, recruits DNA-G. It says, okay, I'm bound now, you can come now. And DNA-G is primase. It will synthesize those first little primers that DNA polymerase needs to get going. Why do we need the primers? Remember, DNA polymerase has to have a 3' hydroxyl to add on to. And we can't, DNA polymerase can't do this just from scratch. It can't be like, oh, well, I can see that there's an A on the other side of me, let me come bring a T. It still needs that previous nucleotide to attach to. RNA, the primase, it doesn't need this. It can be like, I can add wherever I want, whenever I want. So it can come in and make a short little piece of RNA, even though there's no 3' hydroxyl to bind to. So it adds this and gives the starting point for the DNA polymerase to work. So we have DNA ATP binds and encourages all these other things to bind. The first thing is DNA C. DNA C comes in and says, okay, helicase, come and attach. Once the helicase is attached, only then can primase attach and work. All of these have to happen one at a time. If they don't, then the actual structure isn't appropriate for primase to bind. They have to have this one bind first in order for the primase to even be able to fit on there and start doing its job. So it's completed the formation of the replosome when we've added the primer, and DNA polymerase 3 is like, okay, I can start this now. So what does that look like? This is here. So we've got the little replication bubble. The DNA helicase loader, which is the blue one, helps DNA helicase, the yellow one, load. That recruits primase. And once the primer is in, then we can bring in our polymerase. Now, where does the DNA ATP complex bind? It binds in a region of the origin that we call the 9-mer, the 9 unit, which is right here. So the DNA ATP comes in and binds right there at the 9-mer. Then the RNA polymerase binds where it's dissociated. Where we end up forming the replication bubble is the site called the 13-mer. So it's not right where the DNA binds, or the DNA A binds. It's a little bit further down. So DNA with ATP binds and opens up the DNA a little bit down at a place we call the 13-mer. So that sequence is heavy with A's and T's, that 13-mer, because A's and T's only have 2 hydrogen bonds, so it's easier to melt there. Does that make sense? All right. I'm going to move on then. I can't tell if we're just tired, if we're like not understanding, if it's too hot in here. So that's why I've been kind of like checking in with you guys a bit. So once the DNA helicase is loaded, the primase and the polymerase can come in. And we went over this a little bit in the previous one. I just wanted to break it all down for you guys so you had it to study from. Why do we make the primers? Because that DNA pol is stupid and it needs a 3' hydroxyl group to work. So the primase does not need that, so it makes the starting point for the polymerase. Gonna move on from there. After the helicase is placed, though, the DNA A that originally bound to that 9-mer place gets kicked off. It says, okay, we don't need you anymore. Your whole job was to set all of us up. Once we're here, you leave. You're just getting in the way. And this huge, massive complex is formed. We have this guy in blue called the sliding clamp loader, and that helps bring DNA polymerase, which is, it's this yellow one here, and it helps load it onto the DNA. Why do we need this? Because DNA polymerase is kind of like lazy. It's like, well, I did some work. I want to go now. It'll dissociate very easily. So this clamp loader and the sliding clamp make sure that the polymerase stays where it needs to stay and get the job done. It'll keep it from continually falling off and having to be reloaded. That's the whole point of that whole sliding clamp and the clamp loader. We end up loading 4 clamps or 4 polymerases at once, because in our replication bubble, we have 2 places that we're doing replication. The replication fork has 2 strands on this side, so we need a clamp and a polymerase on this side on both of the strands, and the same on this side. So we need to have 4 different polymerases to copy the 4 different parts of the DNA strands that we're looking at. Does that make sense? Yes. Just a clamp loader. We're microbiologists. We're not cell biologists. We think stupid names like Sonic Hedgehog gene are ridiculous. We try to name things for what they are. Makes it a little bit easier for us. Okay. That's all initiation, which of course obviously would take a while because we want to set it up right. Once we get it going, it just goes. So when we elongate on the leading strand, the polymerase just keeps going. because it's going in 5' to 3', and it's like, all right, I'm just going to keep doing my job and catalyzing these reactions and adding more nucleotides. But on the other side of the fork, we have a problem. This end way over here is the 5' end. So as it's making new DNA, the new DNA strand is going the wrong way. And so the polymerase would have to be adding 3' to 5' to keep following. And it can't do that. It has to add from 5' to 3'. So we open up the bubble, it attaches at the 5, where it's going to add at the 5' end, and it's going to copy. Well, when it gets here and it's done, we've now continued and unzipped, and there's a whole area that hasn't been copied. So it copies in a piece, breaks off, comes back to where you've opened it, reclamps, and then copies until it gets to where it just left off. So that's the problem with the lagging strand. It can't follow the replication fork. This one, you see, it's starting at 5' and adding towards 3', so it can just keep going as we unzip. The bottom one can't. So it only can copy what's available. And because of that, it has to keep jumping back to the new area that gets unwound, and that